Hello everyone, I'm Darshana. So in this video, I thought of talking about a topic that is relevant to this COVID situation and COVID vaccinations, as well as I guess it has some relevance in terms of what we perceive as knowledge and some of the relativistic interpretations that can arise even within science, right? So using these vaccine trials and vaccine studies as an example. So to do that, I thought of focusing on this particular research paper first. This particular research paper first. This was published in the New England Journal of Medicine. In This was published in the New England Journal of Medicine. I think sometime back, maybe in April. Uh, so. So this is about a vaccine study. It's not really a controlled trial. I guess it's more like an observational study, which was done in Israel, uh, where so after this Pfizer vaccine was rolled out in Israel, because I think in Israel they had this agreement with the Pfizer company to use that particular vaccine, and I guess it's one of the countries that is one of the most vaccinated countries for COVID. And so this particular study gained some, some traction in the media, and it was hailed as a study to show that, um, it's one thing that the trials show that the vaccines are effective, but this actually showed apparently that um, it works in the real world as well, right? So this particular article, I guess, gained a lot of attention, and actually I came across this article through some newspaper article. So when you read this article, there are a couple of things that I want to highlight here. One thing is that uh, one thing is that the some of the interesting things that they have done in terms of the methodology. Yeah, so now the groups, you typically assume that in a study like this, you have kind of a static group of vaccinated people and unvaccinated people, and you over time you monitor them to see whether what's the percentage of them catching COVID or being tested positive with a PCR test or something like that. But interestingly, in this study, actually they have so the apparently the groups are pretty dynamic. That is, so some of the people who are in the unvaccinated group, uh, they could get moved to the vaccinated group in the middle of the study if they get vaccinated. So the follow up ended at the earliest for following events, and one of the events is vaccination of the unmatched control for vaccinated persons. That is, for each unvaccinated person, there was a matching vaccinated person with the similar in the similar demographic, according to the same similar demographic criteria. But suppose the unvaccinated person gets vaccinated down the line, they could get either removed from the study. I guess they removed them from the study, um, and they won't be considered for later analysis. So it's kind of an interesting thing that they did. Why I don't know why they decided to do that, because usually you'd assume that the groups, the control group and the trial group, would remain static, right? There's another thing that now uh, you are vaccinated, so in the middle of the study, if you have so within 7 days, if you catch COVID, that actually uh, that person would be considered as unvaccinated person, for example. But they were vaccinated, but within 7 days they caught COVID, because the assumption is that it takes 7 days for the vaccine to take the effect. I get the point there, but I mean, then you kind of treat the two scenarios, two scenarios of vaccinated versus unvaccinated scenarios in two different ways. That is, you that could bring some additional benefit to the vaccinated group, because sometimes even if they catch COVID after being vaccinated, if it is within 7 days, you would still consider that person as an unvaccinated case, right? So those are like some minor issues. The main issue that I want to highlight is that it's given in this table, and it's actually like the summary of the study, of all the results that are obtained. Uh, so here we have the progression of, or like the cumulative cases over time, both of the unvaccinated versus vaccinated groups, for both documented infection, I guess, that is if you have some symptoms, and if you are tested positive in the PCR test. This symptomatic COVID, that is it's just based on the symptoms if you do the diagnosis, right? Now, to understand the importance of these results, we have to go back to the original clinical trial data of the Pfizer vaccines, which is published in this particular report. That it's not really published to the, I guess, general public. It's available, but you have to look it up. It's not a research paper. It's just a report that is submitted to the FDA for approval. And there, so it's the same vaccine, right? The Pfizer vaccine. And if you so the I mean, it was reported in the media. This was the initial result that gained a lot of attention in the media, I guess, that this has 95% efficacy. Now, this 95% number was obtained by yeah. So if you look at this particular graph, so you can see how the 95% number is obtained, and and to understand that, we have to go back to how vaccine efficacy is um, computed. So if you go to the CDC, Centers for Disease Control article on how you measure the effectiveness of a vaccine. So this is the formula that is used. You compute the risk among the unvaccinated group, subtract the risk among the vaccinated group, and divide by the risk among unvaccinated group. So it's a relative benefit, right? It's not an absolute benefit. So that's important. We'll come back to the reason why that is important. And so here, if you look at the FDA report, so they had the placebo. Not really sure whether placebo is the correct word to use here, because they apparently use some meningitis vaccine in the placebo group. So you can say it's a control group. And so control group, after 7 days of the second dose, 162 people were tested positive, and they had some symptom, right, of the PCR test. So, and compared to that, 8 people were tested positive and had some symptom. So I'll share all these papers, you can have a look. So 8 to 162, that means we look at the relative benefit. So the 162 minus 8 divided by 162, right, the risk. So it's a very simple approach, and you have 95%, right? So that's how this number was obtained. And the roughly the control group and the vaccinated group were of equal size, that is around 18,000 people. This is the phase 3 trial, I guess, so you have a larger sample. Now, so how they define risk in that particular context is basically the number of people who tested positive after a certain number of days. So I think this 7 days is arbitrary number. I mean, if you say 30 days, that might be different, right? Because that you monitor people for a long time, could be 6 months or whatever. This is an arbitrary number, and the So because the CDC, the CDC definition of risk, they don't really define how to compute risk, right? So it's kind of, there's some subjectivity there, I guess. There's some room for interpretation. So here, basically, they have to compute the risk. They have taken the number of people who have been tested positive after 7 days. Now, if you come back to the original study that I was talking about, the Israeli observational study. So if you apply the same I guess method here, so after 42 days, after the second, I think this 42 days is, I'm not really sure whether it's computed after the initial dose or the second dose, but that is immaterial here. So after 42 days, you can see that the unvaccinated group, they have a documented COVID cases more obvious. There is more. It's easy to understand. So cumulative number of events is the total number of cases, documented cases, right? So unvaccinated groups, 6,100 minus 4,000, 6,100 minus 4,460 equals divided by the risk among the unvaccinated group, right? So we'll take the cumulative number of events because that was the one that was taken in the trial. So that is 0.26 something. That is 26%. That is way below the minimum requirement, which is 50%. If we apply the same thing to symptomatic COVID, cumulative number of events, 3,607 minus vaccinated group 2,389 equals divided by unvaccinated group 3,607. So again 33%, right? So it's way below 50%. Because 50% is the minimum requirement for efficacy. Now, if we follow what I want to highlight is that if we follow the same method that was used for the trial, the observational study, the data given in the observational study would show that the vaccines are not effective, right? But they are not using that method. They are using a different method, and my guess is that the reason for using a different method is to show that it is effective. So it's like you have a conclusion, and then you try to use a method to, maybe they got this data initially, but you use some kind of a statistical method or statistical tricks to somehow show that it's effective, because here they are using something called Kaplan risk. So they are not using that raw numbers to measure the risk. They are using some kind of a more advanced statistical method. And so I'm not really familiar with this risk measure, but this is just a Wikipedia article on how this measure is used. So apparently this is used as a product limit estimator. Like for instance, if you want to compare how a particular drug extends the lifetime of a cancer patient or something. So they are going to die anyway, but it extends the lifetime, and that has a limit. That curve has a limit, right? And so you give it to a bunch of people who are you compare the lifetimes, the percentage increase of the lifetime of the people who receive the drug and versus the control group. So then you get these plots, and you can basically, this estimator will give you a single number using this kind of function, so-called survival function, to compare the two graphs, right? And I'm not really sure whether this, if you look at the theoretical basis of this Kaplan Meier estimator, whether that is applicable in this context, because if you take a vaccine, so the only reason that we have a plot that lasts only 42 days is because we can't practically, maybe they can't gather the data after that, but the plot is defined after that, right? Until the last person, the until everyone who receives the vaccine dies, maybe after many years, when they eventually perish, that then only the curves really end. So, I mean, theoretically, whether I'm not really sure Kaplan Meier estimator is applicable here, but anyway, they applied here. And another interesting thing here is that they use a very small scale here, so that it really, you know, exaggerates the contrast, I guess. So, so I can't really make an allegation here, but obviously, as far as I can understand, what I can say is, if they followed the method that was used in the FDA trial, or the report that was submitted, report of the trial that was submitted to FDA to get the approval, which is a sensible thing to do, right? I mean, you would like to follow the same method. Then, basically, the observational study proves that the vaccine is not effective. But they didn't do that. They used some other fancy statistical method, and based on that method, you know, it's whatever 95% or it's just immaterial. It's a good number. And the media, and in the social media, and usually when we go come across media articles, we basically look at the headline, which I'm also a victim of that. So we usually do that. We don't really go to the source material and check. And the media would say, okay, this was 95% effective in the trial, and now it's maybe 92% effective in the observational study. That means it's good, it's good enough. That's but that's like comparing apples versus oranges, so to speak, because the numbers mean completely different things in these two contexts, right? So that's basically what I want to highlight in this video, that this sort of a statistical relativism, I'm not really sure whether that term even exists, but it's kind of a relativism that is brought about using statistical methods. Because in statistics, I guess there are many, you have some raw data, you can interpret that data in many different ways using different methods. So at least in the vaccine studies and vaccine trials, we see that there's some kind of relativism, and with that comes some kind of a scientific lack of rigor, I guess, because you can basically come up with any kind of method to fit your narrative, right? Fit your objective, fit your conclusion. So that's kind of not very scientific, I guess. I mean, if you think of science as an ideal, uh, the ideal is that there has to be some kind of objectivity. That's why we do these experiments. Cost a lot of money, and a lot of things depend on these experiments, right? The government policies, quarantine policies. Now people are talking about vaccine passports even. Yeah, you have to get vaccinated, otherwise you can't, go about living your daily life. So a lot of things are at stake, um, on these studies, based on these studies. So when you have this kind of relativism, it boils down to opinions, right? The study is basically an opinion with some fancy maths. That's all. Um, there's another example for this. Uh, so this is another study that is about the Pfizer vaccine. So this is regarding the AstraZeneca vaccine. So I think AstraZeneca, now a lot of things has happened since this particular article was published. Now AstraZeneca um, has faced some roadblocks in different countries. I think Denmark has kind of thinking of permanently suspending it. But anyway, we'll look at this article as an example. So here, this was, I think, health secretary or some kind of an official in the UK, and he's citing some study regarding AstraZeneca vaccine, because one of the criticisms that it had was there was apparent lack of data of elderly population, and that's kind of strange thing, because the COVID-19 illness is supposed to affect the elderly people more, and you kind of wonder why there's lack of data for the elderly people, which is supposed to be the group that the vaccine is supposed to help the most. So anyway, and the so there were like good numbers, 80%, 90%, right? That's what was what what the media reported and what the social media reported and all that. Now, if you again go to the actually it's a preprint, it's not a peer reviewed article. So that's another interesting thing. Whenever you see that there's a article that fits the narrative, even if it's a preprint, gets a lot of publicity, but we'll ignore that for the moment. Um, Now here, so again we'll go back to the original paper of the AstraZeneca vaccine, because in the AstraZeneca vaccine, the original paper that was this is one of the key papers, I guess, that showed that it's safe and effective, was published in the Lancet journal. And so one of the things that again they had used the vaccinated versus control group and compared the number of cases 7 days or 14 days after the second dose. And if you look at some of the numbers here, so there's barely any difference. Like for example, hospitalization rates, there's barely any difference between the vaccinated versus the control group. So what they have done is they have used this they have used this robust Poisson regression model. So basically they have mapped the result into a Poisson distribution and then done some comparison. That's how they have measured the effectiveness. So again, it's not the method that the Pfizer vaccine has used. I'm not saying the Pfizer vaccine's method is the ideal, because I think there are some problems with that as well, but it's a different method, right? And I think why they did that was, in some of the results there was not much difference. There was a difference. I mean, the vaccinated group showed you know better results, but the difference was minimal. And so this modified Poisson regression model was used to kind of, then what happens is you kind of expand, or again there's some exaggeration of the difference can result in from that, as I understand. But anyway, important thing is they didn't just go by the raw numbers like the Pfizer trial did. And if you go back to this preprint that which got a lot of attention in the media, the key thing that I want to highlight it now, there's a more important issue here, because here, as I understand now, in all the trials that we looked at so far, the vaccinated group and the unvaccinated group, so they were like static groups, right? Pretty large groups, and they were compared after some time. You vaccinate one group, you don't vaccinate another group. So this is kind of a reverse process. You take the people who come up with a positive PCR test, and you kind of predict which group was vaccinated uh, which group was vaccinated versus which group was not vaccinated. So if I go back to the method, so here it says the logistic regression was used to estimate the odds of vaccination in PCR confirmed compared to those tested negative. Right? So you take the group who are PCR tested positive, PCR tested negative. So it's not just any group of people; it's a group of people who are tested positive in PCR and negative in PCR. And then you use this statistical model called logistic regression model. Sometimes it's used for these data science related problems as well, to predict what is the probability that this person was vaccinated versus the group that was not vaccinated, right? And the So these are some of the features that were considered in the logistic regression model, like whether they are in a care home, geographical area, ethnicity, and so on. Now, there are several problems here. One thing is I don't really understand how you can do a logistic regression, because to do logistic regression you have to have some ground truth. Right? You have to have some um, data that you have validated, where you have the ground truth, you have the correct answer, and you have to predict different things here. You have to predict multiple things here, because you have to predict whether someone is vaccinated and how long, but the time that is um, the time since vaccination, right? And what is the vaccine that they use, because they have compared different vaccines as well here. So I don't know how all those things were predicted, and I don't see the need to use a statistical method to predict whether someone is vaccinated or not, because in a country like UK you readily have access to that data. I assume that they have access to that data. So and yeah, that's basically uh, so yeah, can't find it here. Somewhere it said that the odds ratios were estimated. That is the ratios for those who were vaccinated versus unvaccinated was estimated. Now the problem becomes apparent if you look at this data, uh, because the data set is not balanced. If you look at the positive cases, and out of those, this is the estimation that they received the AstraZeneca vaccine versus the Pfizer vaccine and the unvaccinated group. So you can see that the unvaccinated versus vaccinated uh, data sets are not balanced, right? There are fairly less number of people in the study who are who are unvaccinated. So this is like a completely different method, right? What I want to highlight is that this is a completely different method, and it's actually a questionable method in my opinion, because I don't see why you can't find the vaccinated versus unvaccinated equal sets of data based on the healthcare records or NHS records or whatever. But they have apparently predicted that, right? Because when you do a prediction, there's always an error, and that error can get accumulated, and even that error is not mentioned here. What is the error of the logistic regression model? And there can be all these hyperparameters that can tune the logistic regression model, and that can clearly affect the outcomes. So um, so it's very interesting that they use this kind of a method. Anyway, this is, I'm not sure whether this is still in preprint or whether this has been published, but the message was given to the public, right, through the media, that this is 90% effective or whatever the number, in the elderly population, right? But the important thing is that the method. So again, this problem of comparing the apples versus oranges that you have, you're just comparing numbers, but they mean completely different things. um, yeah, and finally I want to touch upon this Chinese vaccine, Sinopharm vaccine, which is again, the phase 3 trials are not published yet, but phase 1 2 trials are there. It's published, and but still it's approved in Sri Lanka. I guess there's some kind of emergency approval given in Sri Lanka, and apparently there are kind of quite a large number of doses imported to Sri Lanka, and the Chinese nationals are given the vaccine in the first round, I guess. So it's not really clear what will happen to the remaining doses, but something interesting is that the method that is used here is nothing to do with the PCR test, right? It has nothing to do with the PCR test, right? Is basically again, the vaccine is kind of a more traditional vaccine, which is a protein based vaccine, I guess. It's like a weakened form of the uh, SARS-CoV-2 virus itself, whereas the AstraZeneca vaccine is, I guess they call it a recombinant DNA technology, and Pfizer vaccine they call it mRNA messenger technology. So those are like new technologies, brand new technologies, I guess, to get approved. But here this is more like a traditional vaccine. And there the method that I have used is basically they haven't done any PCR test. It's basically they have tested for the antibodies after giving the prevalence of antibodies after giving the vaccine, which I guess it's okay, I guess. But the problem is now, if we, what will happen if they did the PCR test here? My guess is, because PCR test is more like a genetic mapping kind of a test, as far as I understand, because I'm not an expert in biology or any of these things. I'm just interested in these things. But it seems, as you understand, it's more like some kind of a genetic mapping. My guess is, if they did the PCR test here, the vaccinated group would turn would produce more positive cases. That's my guess, and I guess that's why they used the more traditional way of testing for antibodies. So it's again a problem. The issue here is that still the governments, most governments in the world, and in Sri Lanka also, the government uses, the health authorities uses the PCR test as a gold standard for COVID. So what will happen if someone gets injected and if they test PCR positive after the injection? What will happen? I mean, will they get quarantined? No, I don't know. So that's an interesting question, because the still PCR test is being used as sort of a gold standard, even though I'm not really sure whether it can be considered a gold standard, but it's being used. But anyway, the vaccine effectiveness is not measured using the PCR test here. So again, there's some subjectivity. So I want to bring out some analogy here. If we use some analogy from engineering, for example, civil engineering. We all live in houses and sometimes in high-rise buildings, and these are built using concrete and some steel and all these beams and all that. So if a manufacturer wants to select a particular type of cement, or some kind of a concrete mixing method, so there has to be some kind of a method, right, to compare different cement types, different mixing methods and all that, to, because there will be different contractors, subcontractors supplying concrete, and you basically use, I guess, you use the same method. You don't you might not choose different methods when you uh, choose different concrete types in different countries in different uh, times. You try to use some kind of, I think there's some objective method that is there, uh, to some numbers, right, to compare. So that brings some objectivity, right? So you can measure the best or better concrete type, or at least the type that is appropriate for a certain type of building. Now, if you use this kind of an approach there, you use some statistical method to somehow, depending on the situation, uh, to select different kinds of concrete, you will have a really tricky situation there, because you might be living in very unstable buildings. So so it's something like that is happening here, in my opinion, because this I guess it's a even more important problem, because now you basically have no choice. In some countries, you have the passport, you have to get injected, and it has mass vaccination, whether you like it or not. So the studies, I guess there's a lot of relativism there, the way statistics and statistical methods are used. It's basically, the studies are meaningless. It's a strong word, I guess, to use, but it's basically opinions, you know, flavored with some mathematical jargon. That's basically what I see. I mean, it's basically nothing, no better than opinions, and that's a problem, because you might as well just give the conclusion without presenting the data, because one thing that should happen, I guess, uh, from the health authorities or organization like CDC, they have to come up with a more rigorous standard to or rigorous interpretation to measure the efficacy of a vaccine. This current standard is very vague, and when you have vague terms like risk, which is a vague term, right? And you leave a lot of room for interpretation, and that's basically, uh, it's not objective, in my opinion. So it's basically opinions, and uh, it's presented as objective knowledge. That's the issue that I have, because if you are presenting it as subjective opinions, no issue, but the media and the social media, it's presented as objective truth, which it is not, obviously, because of the subjective methods or different methods that are used in different scenarios to uh, obtain a preconceived conclusion, right? So there's a mismatch there. That's what I want to highlight here. And finally, I want to just touch up on this. Even the Pfizer trial, the method that they have used is this idea of a relative benefit. Now, I know that even in CDC guideline, it's basically this is actually the formula is of a relative benefit, right? But now, is that the um, whether that's the um, in terms of vaccinations, whether that's what we really want? Because if we really think about it, the most relevant outcome that we want, or like we are more interested in the absolute benefit, I guess. So here, if you look at the absolute benefit, because this relative benefit, if the really depends on the number of samples in the control group versus the vaccinated group. So if you had 100,000 here, still this would show 95%. If you had 10,000 here, still this would show 95%, which doesn't really take into account the number of samples. So what we really want here is, if we take the absolute benefit here, 162 uh, minus 8 divided by, so it will take 18,000. So this is the number, like it's 0.8%, that is the absolute benefit. If you consider the number of samples in the groups, so the two groups. So and I think, in my opinion, it's the absolute benefit that really counts in some in something like epidemic. But anyway, they have used the relative benefit, and maybe there are some arguments for that as well. So that's basically what I want to highlight, the idea of an emergence of a statistical relativism when it comes to the vaccine trials and vaccine studies. And if you have any other opinions or thoughts about this, feel free to post comments. And thanks for connecting, and see you again with a similar video. Thank you.